Yeah, before we get into that problem, we can we can retrieve from the biobank, from the public databases, from the reference genome databases. Yeah, so yeah, as I just mentioned, the Genome-Wide Association study will report will not report the casual variant. It report the tag SMP or lead SMP or leading SMP. Yeah. So the tag NCMP itself is the representation for a haplotype block for all the SMP in that haplotype block. And sometimes we can use the technique called fine mapping. It's a statistical to further increase the resolution, but still it cannot be increased to SMP by SMP. It's a limitation that if we have more information from the reference genotype, we can calculate the fine mapping for a specific region and try to narrow down the possible SMP, but still we have a set of SMP that can relate it to our phenotype. So we need to remember that the GWAS hit can be read as a region containing something interesting, but not the exact base pair to cause that disease. And since Genome-Wide Association study is just association, so nowadays people will ask you for replication. So you need to replicate in independent cohort. So an independent cohort means that you need to have some independent hospitals, some independent, or maybe some foreign country that can cooperate with you, that they have also a set of patients that have very similar phenotype to your cohort. And you calculate your GWAS result in their cohort and confirm the same finding in their cohort. So that's a major difficulty, major obstacle for researchers outside Europe or outside America, yeah, because we don't have that much independent replication cohort in many countries outside EU region or outside the Americas. Yeah. And the ancientary background needs to be considered. Yeah. So if you try to read the GWAS paper, most of the GWAS paper is contributed from Caucasian people, from Europe, from America. So the GWAS results from European ancientry cannot be directly used in Asian people. Yeah. In African people. There will be some ancientry bias to them. So there's a trend that many countries try to replicate the same GWAS setting from European ancestry. They try to validate their result and construct their own GWAS database in their home countries. And the GWAS analysis result itself is just a starting point. Yeah, we can do further analysis from Genome-Wide Association results. We can calculate the fine mapping, try to narrow down the exact locus, try to narrow down the few SNPs that contribute something to our phenotypes. And we can use functional annotation. If your SNP is located in exome region or in enhancer in promoter, maybe we have a functional annotation that can help us interpret the clinical parameters. And for some region in our genome, we have EQTL. We have the RNA expression data that have been associated with genotype. So we have this kind of database called EQTL. So we can see whether our variant can change the gene expression data profile in relevant tissue. And we can do experimental validation, but experimental validation need to be very careful because we don't know the exact SNP that contribute to our phenotype. And maybe it's a bunch of SNPs. So the experimental design, the functional design must be very careful. Maybe you knock out the wrong SNP and you got negative results. Yeah. And yeah, I will send you my my PowerPoint today. So I think I will not get further detail to the fine mapping or the functional annotation or QEQD. Or so maybe you can Google it, or you can, if you have some question, you can mail me, and we can proceed to the MLD patients. Yeah. So how can we use the Genome-Wide Association Studies results? So after the Genome-Wide Association study, we have the effect size for an SMP and the allowed dosage for each patient. So we have this information after our GWAS calculation. So people will use a technique called PRS, the Polygenic Risk Score. So the PRS definition is pretty simple. For each patient, you select a bunch of SMP based on the clinical findings, based on previous literature. You select a set of SMPs, and you calculate for each patient for their allowed dosage that times to the effect size to the SMP in your GWAS study, and then you summarize it. Yeah, you have a summation for all this calculation, and you got a raw PRS score. And usually people will normalize the PRS score for each patient, so you can rank you can rank all the patients by their rankings. Yeah. And for a patient that had high score, means that he or she had higher genetic risk than average. Yeah. And usually in this study, people will separate this patient into maybe Q1 to Q10. You have 10 group. So for the Q10 group, they are the top 10% in PRS calculation for the whole cohort. So the classic way to build a PRS is that you have the GWAS summarized statistic. So we have for every SMP, we have the effect size and we have the p-value. And because the SMP tend to inherit it in the same haplotype, so we need to do the linkage disequilibrium pruning, so to remove the correlated SMP. And we at this point we often use the SMP that have beta, they have the most largest effect size here and remove some nearby SMP. And we use p-value thresholding to keep SMP in a specific set. That's because if you use all SMPs among your target, you will got something called overfitting. You use too many SMP to define the PRS score. So this cannot be generalized outside your cohort. And after this definition, the calculation is pretty simple. You just calculate for each SMP for the dosage times to the effect size, and then summarize it for every patient, and then normalize the score based on the distribution of the PRS score for every patient. And after 2018 or 2019, there's a new way to generate the PRS. So the input itself is still remain the same. You have GWAS summary statistics, but including the linkage disequilibrium reference panel. And we use modern software like LD-PRED or PRSCS. That software like this can calculate something called Bayesian modeling. So we have a Bayesian probability to shrink our linkage disequilibrium. So we still remove the SMP based on the linkage disequilibrium map, but we can shrink the nearby tiny effect to one leading SMP. So the SMP, the posterior effect for the SMP itself got modified based on the Bayesian model. And then we do the same thing. We use the raw GWAS effect times we use the posterior effect and summarize it and construct our PRS model. So the PRS limitation itself is similar to GWAS. PRS is not a diagnosis. If you have a high GWAS, it means that you have higher than average risk. And how can we define the average risk? The average risk need to get back to the original Genome-Wide Association study, to the cohort that constructing the PRS. You are just having a higher risk compared to the cohort, what the original GWAS did in the cohort. You have average risk compared to the cohort. So this is the second limitation. Yeah, you have a relative risk compared to the cohort, not an absolute risk. Yeah, so still you need to get back to the original cohort. What kind of population can be done for that GWAS? The risk itself is comparing to the, and there will be some population issue, the race issue. So for one PRS that have a good accuracy in Caucasian people may not be suitable be used in Asian people or African people. Oh, I just finished these. So the third limitation for the PRS is that the Asian territory portability is pretty low for nowadays GWAS. Yeah, so the cross Asian territory portability is not that good, and many statistics genetics are trying to do this. And I think in the recent 2 or 3 years, there's a lot of papers trying to combining the GWAS from every population around the world and try to calculate something called meta PRS. Something like the meta-analysis, the meta-PRS. So if you have interest, you can find with the keyword, the meta-PRS, META-PRS. Yeah. So some clinical decisions need to be considered about the phenotype content and disease stage and the treatment options. So a lot of clinical decision cannot be used for parameter used in our association study. So the clinical interpretation itself, I think, is the most difficult part for GWAS study, not the calculation part, because the calculation can be done using modern large language model, using something like ChatGPT or using cloud with proper prompts or proper specifications. You can let the AI to do the analysis, but the interpretation itself is still pretty hard. Yeah, and before we get into the MASLD, we can have a quick recap about MASLD. Yeah, as we talked in the opening of today's lecture, we said that MASLD has around 30% globally for the prevalence. So it's a large discovery cohort can be achievable. And it has a quantitative phenotype, so you can use MRI to quantize their density of fat fractions. So we have a high power for statistical analysis, and the tissue can be accessed. And there's a very important thing that the MASLD itself has a known biology. So for 2 decades ago, someone published a few important genes that are not limited to the PNPLA3, a set of known genes that already known that's highly related to MASLD. So this gene can be used as positive control for GWAS analysis. However, the power for previous GWAS for MASLD is suffer from unknown noises. And still, people are trying to find what kind of unknown noises is. And today's paper, in this paper, they try to find a way to distinguish the subtype in MASLD. So this paper is from Nature Medicine in December 2024. And the title for this paper is the Partitioning Polygenic Risk Scores, identify distinct type of MASLD. Yeah. So the cohort itself is pretty small compared to modern GWAS study. It used only 36,000 of patients from UK Biobank. But the most important thing for that is they all come with liver MRI. So this is the hardest part. All of these patients have both genotyping data and liver MRI. Yeah. And the authors calculated two derived phenotypes from the liver MRI. So he calculated the liver fat content and the inflammation biomarker using the MRI protocol. And it had replication. It do their replication in European cohort from Netherlands and Italy. So they have different cohort to confirm their findings. And they use SMP array plus imputation to do that. And as we just said, this case number is rather low very small compared to modern GWAS study. But the author find a clever way to do the feature engineering. So feature engineering is a term in machine learning field that we can calculate some derived features from our original set of features. So the authors try to have something very important called the VAT. The VAT is the abbreviation for visceral fat volumes. And the authors found that if we use the VAT as a covariate, the VAT itself can contribute most to the MRI's phenotype. The VAT itself has the largest effect size to the fat content and to the liver inflammation biomarkers. And as we can see, the BMI itself cannot contribute to it. The effect size for BMI is near the zero or negative results. Yeah. So the BMI itself cannot act as these biomarkers. So the authors find a very good feature that can have some kind of effect to the liver fat content and to the liver inflammatory biomarkers. So this is similar. This is a Manhattan plot. Yeah, this is a Manhattan plot we just introduced in the previous few slides, but it's in a very fancy way, in a circle way. But it's similar to read that you have chromosome 1, 2, 3 to chromosome 22. So it's just chromosome here. And the author calculated two set of PIS scores. They define two sets of people based on their SMP results, and we will talk it in the following slides. So they construct two sets of Genome-Wide Association Studies here, one labeled in yellow and one labeled in blue. And we can talk about the overall discovery for this paper. The authors found 27 novel loci compared to previous study, using this rather small cohort. So this means that the author used a very good way to define their grouping. So that's why the most important thing for GWAS is the clinical parameter, the clinical grouping itself. And 6 of them got replicated in independent cohort. So the author confirmed these 6 genes have novel association to MASLD. And also, the previous known major loci, including these 5 genes, are all of them are included in the GWAS study. So they are positive control. Yeah, this means that the quality of this GWAS analysis is pretty good. So the title for this paper, the partition PIS, means that the author used a subset of their samples and try to construct different PIS among these samples. So the 2 major groups for the author's definition is the concordant variant and the discordant variant. And the concordant variant means that this variant is associated with the liver fat and also associated with blood lipids together. Yeah. And this kind of SMP will cause systemic metabolic disease, will cause hypertension, cause diabetes, or cause fatty liver. And there's some set of SNP, the discordant variants. These variants will increase the liver fat, but lower the blood lipid. Yeah, so this is the key finding for this paper. Some variants that have opposite effect to the liver fat disease and the blood lipid. So as we described the phenotype, the author named this phenotype called the liver retention type for this discordant PRS. So in this type of patients, they are strongly predicted they will have a fatty liver, they will have an MASLD, and they will have HCC, but they will have the carcinoma. But they are protective against cardiovascular diseases or cerebrovascular diseases because their blood lipids got lower by this variant. And for the concordant PRS group, they increase in all kinds of metabolic related disease. They increase the risk in MASLD, increase in CVD, increase in heart failure, increase in all of this. Yeah. So, and both of these PRS increase the diabetes risk, but they may through the different mechanisms. They also hypothesize, they also have a hypothesis to these mechanisms. So the most important insight for this paper is that there's subtypes for the fatty liver disease. So if we combine these two groups together, because they have opposite effect to the SNPs, so the statistical power will be reduced because you have different genotyping background there. They have different effect for this SNPs to your phenotype, to your fatty liver disease. Yeah. And the author also confirmed this calculation using two distinct ways. So on the left-hand side is the odds ratio. So the odds ratio is calculated from logistic regression. And the right-hand side, the hazard ratio, so it's calculated from Cox regression model, Cox hazard model. So these two statistic method is asking different questions. So the odds ratio means that in the following time among your cohort, the patient had increased or decreased risk for specific disease. So it's just asking about the binary outcome in your following period, the patient have risk for having this disease or not. It's a binary question. And for the Cox regression model, it's asking for time-related question. So during your following period, the probability that your patient have that disease got increased or decreased. So they asked a different question, but their results are pretty similar. Yeah, they both increase in their probability that you will have the disease or increase the probability over time. Yeah, so the author have a hypothesis in their discussion. Yeah, for the previous MASLD drug clinical trial, maybe they enroll patients from different subtypes of MASLD. So because they have different genetic backgrounds, so their response to this drug will be different. So this kind of drug may be the drug targeting for liver retention pathway may not have good enough effect for systemic type of patients and vice versa. So defining the group for the subtype stratification is pretty important for clinical trials or for your study design. Yeah. And also they also confirmed this by comparing their data to the RNA expression data. So we can find that in the discordant group, most of the genes got increased in the liver, only in their livers. So this fatty acid related genes only expressed in their livers. And in concordant group, they have rather less of this gene expressed in their fatty liver. So they also generate an hypothesis graph that for for the discordant group, their liver tend to treat more about they tend to have more fat accumulated in their liver, so that their liver have some kind of function that collect the fat to liver, but with that, the fat content in their blood vessel got decreased by this phenotype. And in systemic patients, their liver all of their blood vessels floated for the fat content, so they floating everywhere, and they cause the disease for most of the metabolic related disease. So there's another paper also in 2024, but it only got published in Journal of Translational Medicine. Yeah, they built the MASLD PRS using exactly the same UK Biobank data set, but it used a traditional way. It just used UK Biobank data set and construct a single PRS, aggregated PRS for all the subjects in UK Biobank. And they have some results, but they does not have any novel result. Yeah, their results are already known. And as you can see, these two groups use exactly the same population, the same cohort for the further analysis, but the result is very different. And that's it only because their clinical grouping, their subgroup stratification way is different. So the clinical parameter itself is the most important part for Genome-Wide Association study, not the analysis part. So for MASLD, you need to collaborate with the hepatologist or to the endocrinologist or to the cardiologist. And if you want to analyze with this stroke, maybe you need to collaborate with neurologists and with bioinformaticians or geneticists to calculate this and to work with this. So we can discuss about the phenotypes for a specific disease and to adjust the statistical model based on the clinical phenotypes. Yeah. Yeah. So I think that's all for MASLD part for today. And I prepare for a few slides to further introduce the whole exome or whole genome or RNA sequencing or the electoral health records analysis. Yeah, all of them only contain around 1 or 2 slides. Yeah, so we have few minutes here. We can discuss about this. Yeah, so as you can see in this summary table, yeah, the SMP array use pre-selected SMP and calculate for Genome-Wide Association study. So it's not suitable for novel rare variants. And whole exome itself is targeting the coding region, the protein coding region, using PCR amplification, using the exome probe, using specific primers to amplify the exome region and sequencing the exome region. So it's suitable for the rare coding variant because rare disease tend to inherit in protein coding way and truncate that protein and cause this rare diseases. Yeah. And whole genome sequencing itself just do shotgun sequencing among our whole genome for a complete discovery. So it's costly, but it's suitable for regulation analysis. You can sequence in the intron region that contain the enhancer, the promoters, so that will regulate the RNA expression. But you need a RNA expression data itself to confirm the results from the GWAS study. So you need to have genome-wide expression data set to confirm the result that you claim. That you claim from your analysis in GWAS or Genome-Wide Association study. You need to directly measure the RNA level. Yeah. And the real world data analysis or the electoral health tracker analysis is another term for is a trending field in nowadays analysis. Yeah. So in a lab setting, in some lab setting, the lab setting is too clean. So your variants got a very good control in your lab, in your cell culture lysate. But in a real world data, it will be pretty noisy. The health record itself, the lab data itself will have some noise among them. So how to calculate and how to analyze this kind of large data need clinicians' input, need clinicians' experience. Yeah. So I have some interesting example here in whole exome sequencing. Yeah, as I just said, whole exome sequencing is containing protein coding region. So it's around 1 or 2% of our whole DNA. So typically you will have 10 to 20 gigabytes per sample. So it's still pretty large. Yeah, so it's still pretty storage consuming or RAM consuming for analyze this kind of data set. So recently, because I'm preparing for some classes, I reanalyze my cohort, and I found reanalyzing a single cohort need around 100 gigabytes of the RAM. So you need to have a higher end computer to calculate this, and it can capture the rare variant and capture some variant that does not get published before. So it's very suitable for analyze for monogenic disease. So this is an example from my work in 2023. Yeah, so at that time I'm in a medical center in Taipei, and we have a very special patient. She got stroke only in her 21 years old. So it's pretty odd for people got stroke in this young age. And the phenotype itself is pretty rare because as you can see in her T2 and DWMI, she has both infarction, she lack of blood flow and hemorrhage. She has blood vessels erupted. So attending physician, also my PhD advisor, she sent her to the CTA, to the CT angiography. And we found that, there's the major vein in our, so this is the major vein. And this vein usually will come all along our school, but it's managed. So it means that her major vein, namely the superior septal sinus, got disappeared, got obstructed by blood clot. Yeah. And because the patient is pretty young, so we have her mother and her father and her grandmother all got sequenced. And we found that she had a very rare disease called factor 2. And the protein region is the protein got the 382 amino acids got mutated from F to lysine. Yeah. And we also confirmed that her mother and her grandmother also had advanced thrombosis phenotype and had stroke phenotype. And all of them have the similar variant to it. So this is how we use whole exome sequencing to determine the phenotype. We found a very specific coagulation phenotype here, and we do whole exome sequencing. And then we find the variant that correlated with coagulation. So this means that the attending physician need to be involved to the analysis. And then we find her patient and confirm the genetic segregation. So we confirm that this is an inherited trait from her family. And yeah, because I have no whole genome sequencing experience, yeah, the data itself is pretty large. Yeah, whole whole exome sequencing is around 1 or 2% of our DNA. So you will have 10 to 20 gigabytes. So for whole genome sequencing, you have 50 times to that data set. So you will need a lot more of resources to calculate the whole genome sequencing data set. Yeah, so for now, I have no experience in it. But I think the most reachable data set is the UK Biobank, because UK Biobank have all of their samples got whole exome sequence. All of their 5,000, half millions of their samples. They have half millions of samples, all of them got whole genome sequence. And UK Bio, you can apply to UK Biobank using your student ID. So you can, so I think it will be pretty cheap for, it will be significantly cheaper compared to us using, applying the data set for UK Biobank. But you need to be, but the data set can only be used in your thesis or in a specific way, not for any publications. Yeah. And the RNA sequencing, as I just mentioned, is measuring the mRNA. So the most important thing for RNA sequencing is that the RNA sequencing itself is trying to capture what the cell is just doing at a specific time point. So the time point itself is very important. So I can share two of my experience. One is that we try to capture the cortisol level from patient, because cortisol is a proxy to stress level. But if you are familiar with cortisol, you will know that cortisol is correlated with our day-night rhythm. So if you don't withdraw the blood from patient at specific time point, cell will get biased from that data. So it's similar to RNA sequencing. Maybe the patient takes some drugs 1 hour before your blood withdrawal, and you will have very different results from the RNA sequencing. So that's the hardest point for RNA sequencing. And finally, the real world data analysis and the electron health I want to share is that this is the recent project I'm working with the Engine Taiwan. Yeah, the Engine asked us to analyze the national biobank result and try to find some genotype specifically in here in Taiwan in our lung cancer group. And I only want to share this data because in this survival plot, we can see that the patients in lung cancer, they have driver gene positive. Yeah, this means that they have some cancer driver gene. They live longer than people that does not have the driver gene. So we are confused. Why people have the driver gene live longer in our national biobank? Yeah. So we discussed with the attending physician. And he come up with the answer in only a few seconds. He said that, oh, these few drivers, all of them have targeted drugs. Yeah, all of these driver gene positive patients receive targeted drug therapy. Yeah, they have targeted drugs. So this is what I want to emphasize, that to interpret something related to the clinical parameter, you need a attending physician, you need a clinical people to help you, to share their hidden experience. They have a sense to interpret the results comparing to our bioinformaticians. So in the paper, they also use SMP array imputation for GWAS. Yeah. So if you want to further analyze their finding, their risk loci, their hypothesized findings, maybe you can have liver RNA expression data and have UK Biobank EHR data. Yeah. because the author used UK Biobank. UK Biobank has RNA expression data, but may not have for the exact same patient, but they have RNA sequencing data. And they have EHR data for them. So you can apply for UK Biobank to analyze the same patient and comparing your result to the published paper. Yeah. UK Biobank is open to every people among the world. So I think that's all for my today's talk. Yeah. So I'm also prepared some take-home figures. So I will send this PDF to Eska so you can see this take-home. But this is only some repeated for my speech, so you can see it by yourself. Yeah. So for now, what time is it? It's 9:30. Yeah. So if you have any questions, yeah, we can have some time to discuss here. But yeah, but I have to claim that I'm a rather shy people. So maybe I cannot immediately have the answer for you guys. So if you have any question, you can mail me, and maybe if I have some time, I can try to. I prefer answering in typing in wording. Yeah.
Okay, thank you so much, Professor Chang, for true and what is a comprehensive presentation. And actually, I have a question regarding the Taiwan Biobank. Is that right? And I heard that from your explanation, the SNP and GWAS is used for rare disease. Is it for recording, like medical record, or prevention of the rare disease itself? That's my question. Thank you, Prof.
Oh, sorry. Please repeat it again. Yeah. What is your question?
Okay, so the of microarray or SNP from the citizen, and is it for the rare disease prevention, or is this for like medical record of the rare disease itself?
Oh, see, Taiwan Biobank itself is faced to the healthy people. So Taiwan Biobank try to have the reference genome among Taiwanese people. Yeah, so Taiwan Biobank itself is collecting data from all hospitals because it's the very good point to connect with the population. But it tried to enroll healthy people. It's similar to UK Biobank. UK Biobank enroll their patient or enroll their subject. All of the 500,000 subjects are healthy during 2005 to 2010. Taiwan Biobank do the similar design. They enroll healthy people for around 200,000 of people here in Taiwan. So they do a comprehensive questionnaire to their where they live, where they belong to, and whether they have a self-reported medical record. So Taiwan Biobank does not have the electrical health record to all our patients, but they construct a connection between Taiwan Biobank data to our national health insurance data. So we can apply that to Taiwan Biobank and try to identify a subset of people. All of them got removed for their sensitive personal information. And there will be a hash ID for us to bring it back to the national health in claims data set. And we can find their medical records in their national health insurance claims. Yeah. So that's how Taiwan Biobanks do here in Taiwan. And also here in Taiwan, there's another cohort called Taiwan Precision Medicine Initiative. So they enroll around 500,000 of people. All of them have some specific diseases. and all of them got informed consent to send their electrical health record to academic clinical. So this specific cohort has electrical health record and genotyping data, but they can be only applied from the hospital that involved in their cohort. So I cannot apply it for now. Yeah, I used to analyze this cohort, but I'm leaving the hospital for now, so I cannot access to that cohort. Yeah.
I see. Okay, thank you so much, Prof, for your explanation. But Prof, actually, I want to confirm again. Do you have like limited time for our tonight discussion? Because I think our member team have quite a lot and quite a lot of things that we want to discuss and consult to you. Is that okay, or do you prefer only by mail or something?
Yeah, you can. Yeah, we can discuss for I think around for further 20 minutes. Yeah, because it's pretty late and I need to prepare for the next day's class. Yeah, we can discuss for around 20 minutes. And if you guys have any question or if some question that I cannot answer immediately, I can mail it back and you can summarize the question list for me and I can find some time to answer it. Yeah.
Okay, 20 minutes then. Will that be okay if we consult about our project, Prof? And I will present our project, and we want to consult about our project.
Yeah.
Okay.
Yeah, it's okay. Yeah, so maybe you can share your
Yes.
slides.
Okay. Thank you, Prof.
Stuff then. Oh, here.
Okay. Before I start, is my voice clear enough?
Yeah.
Okay. Obviously, thank you, Dr. Chang, for the explanations and the material. It was really interesting, and the way that you explained it was very coherent because I remember having questions, and then in the next slide you would answer the question. So I hope that I can give you the same impression with this presentation with our train of thoughts when we are designing this project. So before we start, we are a group of undergraduate students, mostly. We are currently preparing for a competition, iGEM, Internationally Engineered Machines. It's an annual competition which is held internationally, and it compiles a lot of undergraduate students or even high school students or even postgraduates, and they come together and they present their works in synthetic biology and genetic engineering or biotechnology and stuff. In our project this year, we are, as you know, trying to tackle the case of MASLD. Aska, can you continue the slide? Okay. So I don't think I need to explain to Dr. Chang what MASLD is. So basically, our concern was that MASLD is very prevalent in Indonesian population, but the thing is, it's very hard to diagnose. It's a silent disease, and the moment that you start to see symptoms, you are actually on the later states of MASLD, such as cirrhosis or maybe even liver cancers. And there are early diagnosis methods, but the thing is, we assumed that they are invasive for the patients because usually we either draw their blood to analyze their fat content, or we do biopsy, as you had mentioned before, and we come to realize that a lot of Indonesians are actually not comfortable with these kinds of diagnosis methods. They are invasive and they are scary for our people. So what we are trying to build here, what we are trying to design here, is an early diagnosis method which is less invasive than blood drawing or liver biopsy. So before I continue, maybe Aska can display our previous PPT. So we are inspired by this one innovation called the latex agglutination assay. Basically, latex beads are receptors are immobilized on the surface of latex beads, which in turn, when they connect with their targets, are going to cause agglutination. We are interested in this design because, first of all, it is very simple, and we can visually see the difference of agglutination or non-agglutination. So it is easily divined. However, the problem with latex agglutination assays is that every time we want to do this, we have to produce more latex beads. Every time we want to do this assay, we have to produce even more receptors, and we have to treat the latex beads. We have to attach the receptors, and these are laborious. It's difficult to do, and most of all, usually, the receptors are antibodies, and as we know, antibodies are really large. They are large molecules, and when we engineer them into microorganisms for expression, the possibility of misfolding is very high. The solubility issues are very high. So we tried to look for alternatives, and we arrived on nanobodies. Nanobodies are antibodies, but they only contain the heavy chain of the antibodies. So they are single units, and they are so much smaller, but it does not reduce their selectivity on their targets. So what we are trying to do here is perhaps express these nanobodies on the surface of a microorganism so that we can sort of create a latex agglutination beads that are self-reproducible, have lower risk of misfolding, but still maintain their specificity. So roughly, that is what we're trying to do. Okay, maybe Aska can return to our previous PPT. So our project design right now is that, first of all, we are divided into two teams, the dry lab team and the wet lab team. The dry lab team are responsible for analyzing the target bacteria of MASLD, because we realized that liver condition and the gut microbiome are actually related in the concept of gut-liver axis, which was told to us by one of our advisors. So right now, what we are doing is we are going to compile as much bioprojects as possible so that we can identify which bacteria are specific to MASLD conditions. And after we do that, we hope that these bacteria have a specific surface protein or surface antigen maybe that we can use to then predict our nanobody structure, which we will then eventually express into nucleotides and transform into our plasmid and into our microorganisms. What we hope to achieve is that when we transform this microorganism, the nanobody which we have previously designed, predicted in silico, can attach to our target bacteria in that MASLD condition and causes agglutination. Before I continue, is it clear enough, Dr. Zhang? Or
Yeah, I understand what you are talking about.
okay. Okay. So maybe next. Again. For the wet lab itself, we are going to divide our methods into three stages. The first stage is that we are just going to visualize and we're going to confirm that our nanobodies are actually expressible on the surface of the bacteria, not extracellularly. I mean, not expressed and secreted out of the microorganisms, but on the surface, and also that it is folded correctly. For this part, we are going to do SDS page and Western blotting with anti-flag antibodies. For the second stage, we hope that we can start. For the second stage, after we have confirmed that the nanobodies are correctly expressed, we hope that we can try this system into our target bacteria, which are specific to the MASLD conditions. And then on the third stage, we hope to check the sensitivity and the exact maybe limit of detection or the degree of agglutination that happens with each variety of concentration of targets. Okay, next slide. Okay, can you turn to the next page, Oscar? Oh, sorry. Turn to the next page, please, Oscar.
Yeah, we have some internet issue here.
Yeah, I think. Okay. Sorry, so I guess. Okay. Back up a bit, maybe. Page. Oh, okay. I think we'll stop here for the wet lab progression and design, because the thing that we want to discuss to you is actually the dry lab ones. The methods of the design of the dry lab one is that we are going to first predict the nanobody folding, and then after we have the folding, we're going to predict the composition of the receptors. So it's kind of a reverse. Usually people maybe predict the sequence first, but for ours, we are going to do it on the scaffolds first, and then we are going to do the sequence of the receptors. For the receptor sequence, we are going to randomize it so that we can maximize our predictions and our models. After that, we are going to predict the binding energy of the nanobody to the target. And then after that, we are going to use the sequence with the highest binding energies as our wet lab models. But maybe before we go there, does anybody from wet lab wants to continue with the explanation, especially for the part where we have to predict the bacteria target in MASLD, because we're also using statistics methods, specifically PCOA, and right now we're actually coming into a difficult position because we are unable to actually come up with a result that actually describe MASLD microbiome in the gut.
Yeah, that's why I just want to ask, yeah, what exactly for the target for your nanobody? So still any analysis, right? Yeah, I didn't hear any about the exact target you are targeting.
I'm sorry to cut Frederick part. So our exact target currently right now is Listeria bacteria, especially protein internalin A, Prof. And currently we're still confused with our principle of coordination analysis, or PCOA, and we want to actually consult to you about that because we don't get the actual analysis about the bacteria that is specific to the MASLD patient itself. And after this, we want to present you the data that we have. And is Tirdi here? Can Tirdi present the data from yesterday discussion? Tirdi or an other dry lab member?
Okay.
Thank you.
Excuse me, Aska, I can represent the dry lab team. Okay, I'll present our dry lab works. Okay. So initially, we are still trying to figure out how to find the biomarkers of the MASLD. So here's our pipeline of finding the biomarkers of the MASLD, starting from the 16 RNA, and it goes to the chyme before it generates into the OTU table that we are going to use. And we are currently facing a challenges where, as we mentioned before, the PCOA analysis is actually show a bad sign, because as we can see right here, a different country might have a different characteristic of the bacteria based on this PCOA, and based on this PCOA, a different country might have a different signature bacteria. So I'm going to ask specifically about the best approach of the finding the biomarkers of the MASLD, because if we look at the PCOA, it is quite concerning because each country has a different characteristic.
Yeah, but maybe you can focus on the dominant strength in your country. Yeah, because it's a very suitable topic for agent test. Yeah, you try to find a way to figure out something that can solve the problem in your home country. Yeah, so maybe you can try to find the dominant strength in your home country first and focus on that strength, or some strengths that have similar response in your PCA analysis. Yeah. But I'm still confused. You mean that the E. coli itself will accumulate in our liver, or so what exactly you are targeting about? So why you are analyzing from the strain of the E. coli? I still cannot get your point.
So as far as I'm concerned, E. coli acts as a biosensor, which will target the target protein of the bacteria that we will targeted in the MASLD disease. So in the E. coli, we will find the nanobody, and the nanobody itself will be generated with a deep learning, so with a computational helps that will attach and will agglutinate to the target. And actually, we did some analysis in terms of the signature bacteria itself, and we have found that some of the project actually has a similar, I'd say, about the signature bacteria, but we don't know for sure if it's well characterized or no. So that's the problem. So as we mentioned before, we are currently using Listeria because it is well characterized, and we can design the nanobody straight away.
So you mean that for they have different profile in their gut microbiome, and you want to try to develop some nanobody to target the specific bacteria in gut as a proxy to MASL.
Yes, exactly.
Oh. Yeah. So still, I think you can focus on the dominance trend in your home country, or if you don't have that sample, maybe if you have the budget, you can do the metagenomics analysis for maybe a few samples. And try to capture the dominant and try to compare with the open databases. And you can focus on maybe there's some dominant strand that are different in MASLD people and healthy people in Europe. So I think this is a very specialized question. You can be only answered country by country. Yeah, so you need your local sample or local data.
Yes, Dr. Chang, exactly. So from the PCOA results, also we can see that each of the groups are actually clustered exactly by their origin of countries. So maybe can you share your insight on regards of the GWS, which you also said that the dominant population of the data is actually Caucasians, and maybe you can share how your experience on completing those missing data.
But yeah, there's no way for the missing the data. Only if you have enough of different. Yeah, but with the PCA, I think maybe you can try to set the country as a batch effect to minimize the effect from the trend from different countries. But if you set the country as a batch effect factor, maybe all the signal will get lost. Yeah, I'm just saying that maybe you can try this, but probably maybe you will get something that contain all the noises, but still you can try that. You just put the country as a battery effect and then remove the battery effect, maybe use it as SMA, yeah, using the specific library from Python. Yeah. Maybe you can try that, yeah, because dry lab analysis does not cost any money. Yeah, so yeah, it's pretty late for now. Yeah, so any questions, or you guys can write your question and ask to mail me. Yeah, so maybe we can discuss through emails. Yeah, and I can share my Gmail to you, so you don't need to mail to my institutional mailbox, so it's very hard to use it.
Yes, it will be a pleasure. Thank you, Dr. Chang.
Yes, I think that will be perfect. Thank you so much, Professor Chang, for your insightful knowledge and for sharing your time here with us. And before we conclude our today's meeting, can we have a documentation first, Prof. Chang?
Yeah. So you need to share the video, right? Or
Yeah.
yeah. Okay.
Okay.
Yeah.
And the others, the team member, please turn on your camera. Okay. Our documentation will be led by me. And in 3, 2, 1. Okay, once again. 3, 2, 1. Okay, thank you so much, Prof. Chang, for today. We hope see you soon, maybe in the future, and good luck for your future endeavors. Thank you so much, Prof.
Thank you. Yeah.
Thank you, Dr.
Yeah.
Thank you.